Howdy. Today, I'll present BoneMet, the first open, large-scale dataset for AI-driven research on breast cancer and bone metastasis. Let's start with why this matters. BCBM is like finding tiny leaks in a vast water system. Let's see, the bone are the pipes and the metastasis are the leaks. Current tools like X-rays or CTs or low-resolution sensors that miss the small leaks or the early metastasis and expose patients to radiation. By the time leaks are detected, bones are severely damaged, cutting the 5-year survival rate by almost 20%. This leads to the classic catch-22 problem. With the clinicians being hesitant to image due to the radiation effects, we cannot feed and train AI models for better diagnosis. Training AI for BCBM is like crossing the desert to reach the oasis. The oasis represents accurate diagnosis, but today's datasets are like small, scattered puddles that do not provide enough data for the journey. The problems include human datasets, that is low resolution and sparse, brain models, noisy, inconsistent, temporal blindness, no progression tracking, and modality silos cannot combine X-rays with CTs or lab data. Without the BoneMet's OASIS, a 67 terabytes of multimodal dataset, AI models collapse from overfitting before reaching solutions. BCBM, or the breast cancer bone metastasis, occurs when breast cancer spreads to bones, creating a vicious cycle. Tumors weaken bone, and bone damage fuels tumor growth. Patients face fractures, pain, and lower survival rates. In order to increase faster clinical research for this, the BoneMet dataset was created. The BoneMet is a 67 terabytes of imaging along with biological data, the unique portion of this dataset to train AI for early detection. The BoneMet dataset is a murine dataset, murine being mice. But why mice? Mice allows for controlled studies. The main issue of radiation is completely elevated when using mice. Mice allow for weekly radiation scans with less than 0.4 level of radiation, which is completely impossible in humans. Unlike other fragmented datasets like INSPECT or MEDPICS, BoneMet offers three unique features. One is the multimodality, involving 2D X-rays along with 3D CTs with the biological records. Second is the longitudinal tracking, weekly scans over a period of 3 to 5 weeks once the breast cancer was inoculated. Third is the APIs, automated segmentation and registration for reproducibility and replicability to other cancer problems. This BoneMet dataset was created using 6 steps. As you see from this image here, this represents the BoneMet pipeline. First is the rotational X-ray imagery, which is captured by the sky scan, as you see here, hardware, starting from the beginning of the breast cancer inoculation to the end of sacrifice of the mice for 3 to 5 weeks. With the step of 0.8, a total of 260 X-ray projections for each scan and covering a total rotation of 208 degrees is collected for each of the mice. The total scanning process for each mouse takes around 10 minutes. Once this is done, the 260 X-rays are then reconstructed into the reconstructed CT imagery, followed by which the segmented CT imagery involves segmenting only our region of interest, the tibia, and the fourth, after segmentation, each tibia is registered with their reference in order to maintain the spatial temporal information. After which, we only focus on our region of interest that has the BCBM metastatus, that is the metastatus, and they are cropped and set aside along with the biological information, that is the mice medical records. This includes records for individual mice regarding the animal ID, strain, date of birth, experiment data, tumor inoculation, treatment type, treatment regimen, etc. All these 6 steps provide us the entire 67.87 TB of BoneMet dataset. All the images are stored in the PNG format, while the last one, that is the numerical data, is stored in the CSV format. Now, 3 main softwares were used for this dataset. One is the NRECON, which is used for reconstructing 260 X-ray projections into 3D CTs. Second is a SEG API for segmentation, isolating only TBA as we saw in step 3 by keeping a threshold of 75 pixels in the grayscale imagery of 255 pixels, 255 pixels. Third is the regist API or the registration, which aligns the reference along with each of the TBA to maintain the spatial temporal alignment or the STA. Second, experiments to demonstrate the applicability of the dataset were carried in 2 different data. One is the 2D X-ray images and second is the 3D CT images. Different models like vision transformers, CNN, and generative models were employed for this regard. The hardware used is, as we saw from our pipeline, the SkyScan 1276 was used to get the X-ray imagery, and the hardware involved using NVIDIA RTX A6000 GPU, which has 40 GB of VRAM. Now, as we saw in the previous slide, 2 different kinds of training took place. One is using the 2D X-ray images, and second is the 3D CT images. Now, let's divide the results into 2 different divisions. One is the diagnosis and other is the prognosis. With respect to diagnosis, the images were 2D X-ray and 3D CT. For the 2D X-ray, to find out the applicability of the rotational X-ray dataset, 2 models were used. One is the vision transformer variant swim base and other is the spatial temporal swim base. And one is with respect to the 2D, the vision transformer with STA provided the higher accuracy, 89%, than the vision transformer without the STA, that is the spatial temporal alignment. This happens because the STA reduces overfooting, which is 6% with STA, then compared to 20.4% in without STA. And the vision transformer swim-based variant was applied to the 3D CT images, which produces 92% accuracy, then compared to the other CNN-based models like the bit transfer and also the efficient net version version 2. That this is because the volume information is better aggregated by the self-attention in spin base than compared to other CNN models. Now, with respect to prognosis, we have 2 different types. One is the use of generative models and other is the conventional CT reconstruction. With respect to prognosis, since our main focus was on the reconstruction of CT images and how well the bone information is being featured, we used 3 generative models. That is the 3D generative adversarial network 3D GAN. The second is the temporal variational autoencoders TVAE, and third is the spatial temporal variational autoencoders, which were mainly used to get the bone prognosis in the 3D CT reconstruction. We find that the spatial temporal variational autoencoder provides the best SSM, the commonly used metric for the 3D reconstruction. That is at the level of 0.860, then compared to 3D GAN and also temporal variational autoencoder. The second is the conventional 3D reconstruction methods, which requires 2D X-ray images from multiple angles like we did in the first step. Two different kinds of NERF methods were used. One is the pixel NERF and other is the MED NERF to reconstruct 3D CT scans. We find that the PSNR and SSIM for MED NERF is better than compared to pixel NERF, as you see here, 30.2 PSNR and 0.81 0 for SSIM. This allows for this allows the MED-NERF method to produce high quality reconstructions, which demonstrates the applicability of the BoneMet dataset for sparse angle CD reconstruction, that is the conventional CD reconstruction. Now, one of the main or the central idea of this paper is allowing the dataset to be open source for replicability and reproducibility for both the BCBM and also other cancer applications. So the BoneMet is open access. It includes the 67 terabytes of dataset hosted on Hugging Face, and it also provides GitHub tutorials and a Python API PVPA packages, and it is also ethics approved. The URLs are given here to further see more on this open source and hosted BoneMet dataset. Now, let's look into how we can translate this murine dataset into the human dataset. First would be human validation, testing models on the datasets like inspect. Second is expanding sites, that is adding spine metastasis because the current dataset focus only on TBA or femur. And third would be the multimodal fusion, merging images with the EHR or the biomarkers for earlier detection. Now, let's look into what are the challenges that we find from this paper. One is the mouse human gaps, that is, the disease progresses faster in mice than in humans. Second is the manual steps in processing, that is, fibula or tibia cropping slows scalability. And third is the clinical hurdles. Human radiation protocols differ than in mice. Now, to summarize, the BoneMet fills the data scarcity gap for BCBM AI. It achieves 92% accuracy in diagnosis, and it also provides open tools to accelerate research, and it also helps in the foundation of multimodal ML models and safer humor diagnosis. While I was reading this paper, I found a potential for better multimodal ML models and analysis. So I have done a little bit research to further understand this. Now, with respect to BoneMet being applied for multimodal machine learning, we find that the BoneMet's multimodal data could be used to pre-train foundational models, that is using large language and vision information medical models for the bone metastasis, or to predict biomarkers from imaging, that is using that can be done as a future expansion, or using generative AI models to synthesize data to fill gaps, that is the spine metastasis or the spine CTs. So, but the challenges would be is to get the spatial temporal information like we did with the murine dataset, that is the vision transformer with STA and without STA. That would involve getting weekly human scans and also all the biological data like we did with the murine, this taking its individual ID, the strain, the date of birth, the treatment type, treatment regimen, etc. But if we could do this, applying BoneMet for the multimodal machine learning analysis would provide further SSIM and accuracy than compared to other models that were used for in both prognosis and also the diagnosis. I would like to thank Professor Dr. Yang Shen for giving me this hint on using multimodal machine learning for this wonderful dataset, and also the original paper authors for this groundbreaking work. And I would also like to acknowledge the AI tool that I used, ChatGPT, in helping me refine this PPT, and also internet sources like Wikipedia, Google, and others from which I have taken the images and the concepts. Thank you. Let's discuss how BoneMet could benefit our work. Special thanks to the authors for this pivotal resource. Thank you.